Today let us study about the morphology of chromosomes, like how the chromosome structure exists and where we can see this particular chromosome clearly. The chromosome appears in a rod-shaped structure and they are the dark-stained bodies. They were first discovered by Wallier in 1888. Each species has a different chromosome and it can be denoted in terms of number, that is 2n. 90 somatic cells contain 2 copies of each chromosome which are identically morphed. So those chromosomes are known as the homologous chromosomes. Coming to the morphology of chromosomes. During cell division, the following structural features can be seen under light microscope only. They are like chromatid, centromere, telomere, secondary construction, satellite chromosomes. Chromatid. It is a structural and functional unit of chromosomes. It is seen in metaphase and the chromosome appears longitudinally divided into identical copies, which is known as chromatid. The chromosomes of a chromosome appear to have joint pillar and pin called centromere, and this is the connection between two arms of a chromatid. The two chromatids making up a chromosome are produced through replication of a single chromatid. That is, so they are referred to as the sister chromatids because they are appearing in the same morphological features. In contrast, the chromatids of homologous chromosomes are known as non-sister chromatids. Here we can see the non-sister chromatids in clear. Now let us study the centromeric region, that is centromere. The region where 3 sister chromatids of a chromosome appear to be held together is called centromere. As we all know very well, this can be seen only under the light microscope. Centromere generally appears as a constriction in the chromosome. Here it is also termed as primary constriction, where the division of chromosome network takes place. Chromomeres are first part moving towards the opposite poles during anaphase. That means the chromatids can move towards another directions. The remaining regions lag behind and appear, and if they were being pulled by the centromere. Therefore, chromosome movement is due to the centromeres of chromosomes, hence they are also known as kinetochores. In most cells, each chromosome has a single centromere in a fixed position which does not change except due to structural chromosomal aberrations, which means the changes that takes place in the structure of a chromosome. Therefore, the position of centromere serves as an important landmark in the identification of different chromosomes of a species. With this, we can differentiate the chromosomal types. Each chromosome is divided into 2 transverse parts by its centromere. These parts are called as arms, as we have studied earlier. Now let us learn about the types of chromosomes. Based on the position of centromere, actually we can study different types of chromosomes. Sometimes the centromere can be away from the arms. Sometimes the 2 arms are distributed equally from the centromere. So like that, we have different types of chromosomes. One is metacentric. That is, the center of chromosome having equal arms and appeared as V-shaped during anaphase. The second one is submetacentric. Centromere is on one side. It means no the centromere is distributed equally to the 2 chromatids. Second one, acrocentric. So the centromere is located close to one end. It means no centromere can be pulled opposite to the another arm. Last one, telocentric. Centromere appear to be at one end of the chromosome. We can see the different types of chromosomes here structurally. The first one is telocentric, second one is acrocentric, third one submetacentric, and fourth one metacentric. The long arms are known as Q arms in general, and the short arms are denoted with P. Coming to the telomeric position, how the telomere can be seen in a light microscope. The 2 ends of a chromosome are called telomeres in general. They are highly stable and they are not able to fuse with each other. It is generally accepted that the structural integrity individuality of chromosomes is maintained due to telomeres, and the all stable chromosome ends are composed of telomeres. That means the telomere makes the chromosome stable if and only the secondary construction takes place. That is when we can see the sad chromosome structure. Here we can see the nucleus content, how it can be seen in the telomeric region. Now let us study about the secondary constriction. As we all know very well, the primary constriction can form the stable chromosomal structure, and but now technically if the secondary constriction takes place, then only the stability comes to the telomeric position. The chromosome which have the telomeric position is structurally and functionally stable. In some chromosomes, a secondary constriction in addition to primary constriction is also present. It is called secondary constriction. It is present in short arm near one end, or in many chromosomes they are located to the long arm nearer to the centromere also. The region between secondary constriction and the nearest telomere is called satellite. Chromosomes having a secondary constriction are called satellite chromosomes, as we have discussed earlier. It is denoted by SAT sat. The number of sat chromosomes in the genome varies from one species to another. Like the humans can have this number of satellite chromosomes, and the other organisms like plants and animals can vary from the human number. Human somatic cells have 10 sat chromosomes. That means each human is having individually 10 sat chromosomes in a chromosomal type. Nucleolus is always associated with the secondary constriction of sat chromosome, whereas the nucleus is associated with the first constriction, we all know very well. Therefore, secondary constriction also called the nucleolus organizer region, that is NOR, N-O-R. Sat chromosomes are often referred to as nucleolus organization chromosome, and this can also be known as nucleolus organism chromosome, NOC. NOR contains several hundred copies of gene coding for liposomal RNA. When if there is need for the coding of proteins, yes, we need number of RNA. For that coding RNA, we can generally use the NOR regions which code for rRNA formation. Yes, we can see the secondary constriction which took place and the satellite formation here in this slide clearly. Here we can see the morphology of chromosomes, the telomeric position, two sister chromatids, secondary constriction where it is taking place, and centromeres, that is formed by primary constriction, and the short arm, long arm, as well as the satellite. Thank you.